All right, and we are live. Hello, everyone. Welcome. I'm Raven, and today we're having a conversation about the new Lancet paper targeting the SARS-CoV-2 reservoir in long COVID. If you're a patient, provider, or researcher, you know that long COVID is complicated with at least 10 major drivers identified, and these are covered in our IACI provider manual releasing this month. So, very exciting. Be sure to follow us for that information. But those drivers include things like persistent pathogens, immune dysfunction, blood clotting issues, mitochondrial dysfunction, and more. But today we're focusing on one of the most urgent questions in the field, and that is if the SARS-CoV-2 virus itself is persisting in the body and driving symptoms, how can we target it to treat long COVID? So to get started, we should clarify what we mean by a SARS-CoV-2 reservoir. How is it defined? And what evidence is presented to support the notion of a viral reservoir? Amy, as the lead author of this paper, can you further elaborate on what a reservoir is in this context and what the strongest evidence is for persistence right now?
Sure. So a reservoir refers to a small amount of a pathogen, so a viral organism, bacterial, even fungal, that persists in someone's body over time and could, in that sense, in this small reservoir, drive inflammation or other issues that could cause symptoms. And when we're obviously studying reservoirs in patients with long COVID, whose cases began with infection with the SARS-CoV-2 virus, what we're looking at basically when we're talking about the straight SARS-CoV-2 reservoir is the straightforward possibility that some people who develop chronic symptoms after getting a COVID infection might just not have fully cleared the virus from their systems. And instead, the virus persists in one of these reservoirs, which often, and again, this is an ongoing field, so there's a lot of research that still needs to happen and that is still coming in. But so far, we do think that these reservoirs are often localized to tissue. Part of that is because SARS-CoV-2 as a virus infects every major human tissue type. So it can spread throughout the human body greatly during acute infection. And thus, if it persists, it likely can persist in many, many, many different body sites. And also, to some degree, persisting in tissue allows pathogens in a reservoir to evade the immune system a little bit better than they might do if they were, for example, just free floating around in someone's blood where there's a lot of immune cells and they might get more recognized and killed. So that's basically the SARS-CoV-2 reservoir. And now evidence for SARS-CoV-2 reservoir and long COVID comes from a number of studies, all of which actually use different methods in different cases to try to establish if someone fully hasn't cleared the virus. And some of those studies are teams that have actually collected tissue samples from patients with long COVID. So in order to get at this tissue-based reservoir, what teams do is do biopsy procedures where they can collect at least some forms of tissue, for example, from the gut or from the lymph nodes or from the olfactory bulb of the nose, let's say, from people with long COVID, and then look for the virus, its genetic material, and its proteins in those tissue samples. And so you'll see if you look at the paper, we actually have a table in the paper that shows that teams have found SARS-CoV-2 long after initial infection, so sometimes hundreds of days after someone was infected, in tissues such as the lining of the gut, in the olfactory bulb of the nose, and in other body sites or tissue collected from patients with long COVID. So there are some, of these tissue biopsy studies that show that there's persistent RNA or protein from the virus after acute COVID. Then there are other teams who use the immune system as basically a biosensor of SARS-CoV-2 persistence. So what they'll do is they'll look at the activity of, for example, T cells, which are cells that circulate throughout our tissues, and they can sequence the way that those T cells are behaving to determine if they're still responding to a virus like SARS-CoV-2. And if they're in that state, it suggests that the virus is likely still there. So there's some teams who have shown that immune cells in patients with long COVID still seem to be responding to the virus, which suggests persistence. And there's last studies that have looked in more kind of what we call accessible fluids, such as blood, for proteins created by the virus, which like the spike protein, for example, because even if the reservoir, the virus itself, for example, could be in someone's tissue, let's say in their gut tissue, it's possible that the spike protein sort of buds off the virus in a little vesicle that we call an exosome, where it could get into the blood. And the thing there is that it's probably or likely tiny amounts of spike protein that might leak into the blood from these reservoirs. So if you have a very sensitive technology, which means that it's able to find tiny, tiny amounts of protein, you can likely try to pick that protein up in circulating blood. And so there have been a number of studies now using these ultra-sensitive methods to find spike or other SARS-CoV-2 proteins in blood or other fluids from patients with long COVID that do show circulating proteins, which could also be indicative that patients haven't fully cleared the virus. So that's some of the evidence.
Thank you, Amy. And so as far as what we can you talk about? Did I cut out? Good night. Okay, so as far as the biological pathways for how the virus can persist in the body, Amy, you've already talked a little bit about them, but I just want to kind of make them a little more explicit for the listeners. In the paper you identified, it was very helpful graphic where you outlined 3 different mechanisms for how persistence might happen. So can we talk about those mechanisms a little more in detail?
Sure. Yeah, so exactly. There's a figure also in the paper where we draw basically 3 different, types of persistence, if you will, all of which could be happening in patients with long COVID. And we have a number of studies underway to try to figure out which form of persistence might be occurring in certain patients. But the first one is that the virus persists, again, let's say in someone's gut tissue, and it basically persists as inert RNA to some degree. Exactly. So it's not really creating new versions of itself or new proteins. It's just RNA that continues to persist. It could still provoke the immune system, but it's not creating any proteins or new viral particles. Then the second one in the middle, the second scenario for persistence, is that the RNA of the virus persists, let's say in tissue, but does go through periods where it produces proteins. It's called translation. It makes proteins like the spike protein that might, for example, like I said, leak into blood, but it's not actually creating full new versions of itself and fully replicating. So that's called, it's translating, but not replicating. And then in the last scenario, the virus is replicating and creating new versions of itself, new virions, and those new versions and virions impact new cells, which can actually potentially make the reservoir, move from cell to cell and even increase over time. So those are the 3 types of persistence. And one of the key reasons we delineate those in the paper is because if you're trying to treat the SARS-CoV-2 reservoir with different combinations of drugs, antivirals, or monoclonal antibodies, or immunotherapies that could support the immune response, for example, you have to think through those different ways that the virus might be persisting, because the way that drugs might work to target the virus could differ based on how the virus is acting. Because, for example, an antiviral, for example, Paxlovid, inhibits certain parts of how SARS-CoV-2 replicates. So if SARS-CoV-2 isn't necessarily replicating, if it's not in that basically third scenario state in that diagram, Paxlovid might not be actually very effective in clearing it. So we have to think when we're using therapies in long COVID, we have to try to understand what the virus is doing to be able to most successfully get rid of it.
Thank you, Amy. And I'm glad that we were able to pull up this graphic. Sorry for the confusion as I figure out how to maneuver this software. But so going back to what you were saying about actually targeting it, to target it, we would have to, I guess, detect it, right? So what technologies do we have to potentially detect a viral reservoir, and where are we, I guess, in a clinical sense, to actually putting some of these therapeutics into practice that we're going to talk about later, the potential therapeutic approaches?
Yeah, so you don't actually have to detect it to try to treat someone for persistence. You could assume it's happening and try to treat them. And I'll be honest that, for example, there have been some trials of Paxlovid to try to treat potential viral persistence in long COVID patients. And those trials were somewhat designed on the hypothesis that some of the people in the study would have persistent virus. They didn't actually recruit people into the trials based on actual measures of persistence. So just something to keep in mind. But we do in this paper make the argument for the fact that we think that trials to target persistent virus moving forward should try very hard to try to measure something to show that a person entered into a clinical trial for some kind of treatment to try to get at persistent virus really does have persistent virus. For that reason, because obviously if the goal of the clinical trial, which most trials is for the group to respond as a whole to the treatment, in other words, for the trial to meet its primary endpoint, you do have to make sure that enough people in the trial have the issue of persistence that the drugs are trying to target, or the results of the trial could be negative. So there's different ways of looking for a reservoir in people. You could do those biopsy studies, right? Where you could get a piece of tissue from someone and look for the RNA or proteins from the viruses in those tissue samples. But those biopsies have downsides because they're invasive to do, they're expensive to do, and they just require a lot more time and effort. So to biopsy someone in order to put them into a clinical trial is definitely a big undertaking. So what the field in the clinical trial space and persistence could very much benefit from is our more accessible diagnostics of SARS-CoV-2 persistence. So something that can be measured in an accessible fluid like saliva, blood, or even stool, right? And so those tests come into either looking for the RNA of the virus, for example, in host cells, where, for example, the RNA might not be free floating in the blood, but it might be persisting in a platelet or inside a macrophage immune cell. And maybe if you can find that host cell associated RNA, you'd be able to capture that from circulating blood. Or, as I mentioned before, there are those tests that can find the spike protein or other proteins. And maybe you can use the identification of those proteins as an inference that someone has a reservoir, and you could use that to recruit people into persistence trials. Or, like I said before, you also have some of those new types of tests that people are increasingly using to use T cell responses or shorter-lived plasmablast immune cell responses to indicate immune system response to persistent virus, which could also be used again as a biosensor of persistence that could allow for recruitment into trials. But that's what a lot of we talk about in this paper, the long COVID field right now needs to really iterate those diagnostics more. There are teams working with these different technologies that can potentially identify the virus or its proteins in these accessible fluids. But we need to better understand how the different possible diagnostics for identifying virus or protein perform against each other and how well they really reflect the presence of a reservoir in tissue. So a lot of our efforts moving forward are trying to do programs to better understand that scenario.
Thank you, Amy. And David, on that note, knowing that pathogen persistence is a driver of IACIs, what are the challenges for providers who are interested in investigating pathogen persistence?
Yeah, I think to echo Amy's, really well laid out statements about, persistence and some of the targets around persistence. When you translate to the clinical side, the first thing that we need is a reliable method of identifying persistence. We currently don't have it. And I think that a huge call to action from government bodies is that there is sufficient evidence now that any reasonable person can understand that persistence is a part of the disease process of many infection-associated chronic illnesses. And that necessitates an investment in our ability to detect these things. We need to be giving physicians tools that are easy to reach for, that allow them to identify persistence in their patients. The next huge area of research that would decrease the uncertainty around treatment is understanding how to target reservoir, how to target persistent pathogens in unusual places. Most of the antivirals, most of the monoclonals that we have are really designed for targeting the virus when it's, as Amy was saying, circulating in the blood. And they're less designed, and that's not all of these drugs, but most of these drugs are less well designed for targeting persistence in tissue, targeting persistence in immune privileged sites, targeting persistence where maybe it's not where we think of these pathogens usually being. And so to that end, we often don't quite know how to, often don't know how to prescribe medications, that are trying to target pathogens where they're not supposed to be. So we need a lot of additional research that allows us to better understand, okay, well, you used Paxlovid when there was, active acute infection, but when should you use Paxlovid in the context of chronic infection? To that end, as we're thinking about clinical translation, that's when we need to start to think about research that can support responder analysis. As Amy said, many of these trials are going to fail to meet their primary endpoint in the whole population. But we're expecting that because it is, long COVID is not yet a very well characterized diagnosis. And we need to be able to understand who's responding and why to different drugs. And that's going to help us to really understand who gets what drug and when. It's also going to help us to understand combination therapies, which are also going to be really important here. The combination of an antiviral and a monoclonal, the combination of multiple antivirals. This is how many other illnesses that involve chronic infections, such as HIV, this is how treatment has developed there. And we need to learn from this playbook and move forward, not just with repurposing of old drugs, but the development of new therapeutics. But overall, when clinicians are thinking about these things, I think we can't say enough. Folks who are coming to these clinics, to long COVID clinics, they're extremely sick. And often we can't just be throwing drugs randomly against the wall because each drug is not without risk. Each drug can take their toll on a person as they're trying them out. And so, what's much more important is that we have, really good quality science and well-designed trials that are targeting persistence to inform a clinician that is keeping up with the evidence.
Thank you, both of you. And so that brings us to our next topic, and that is the potential therapeutic approaches to target the reservoir. And I'm going to throw this graphic up on the screen. All right, here we go. So we're going to spend a good chunk of time here on this because there is a diversity of options that you presented in the paper, Amy and team. And I'd love to look through these potential therapies. The paper covers things like antivirals, cytokine-based therapies, next generation vaccines, monoclonal antibodies, and more. And each of these work in their own different ways. So what are the considerations for these approaches? We probably don't even have enough time to go through all of these in extreme detail, but when you look at this figure, what are the main takeaways and what do you think stands out the most that you want everyone to know?
Yeah. And what was actually cool about writing this paper, and obviously David's on the paper as well. And I think there's actually 34 authors on the paper. And, we're part of a long COVID consortium, basically, that PolyBio has organized. And so all 34 authors on the paper are studying SARS-CoV-2 persistence in various capacities in patients with long COVID. And many of them are some of the trialists who are people who are trying to run trials of some of these different drugs. And so what was cool is even in making this figure, we worked a lot as a group to hash it out. So it does represent a lot of people in the fields ideas coming together in terms of finding some consensus, at least, of where we're at. And, okay, yes, there are luckily a decent number of options that you could use to target persistent virus. I think the biggest issue that we face, though, that I'll just say off the bat, is that not all of these therapies either exist or are able to be used in long COVID. So only certain drugs are actually approved and out there to be used. So for example, the first type of drug, which I think would be straightforward to people that we can trial to try to target persistence virus, are antivirals. And right now, Paxlovid really is the only antiviral that is out there that can be sort of, mixed and matched into trials without needing the drug company to participate and potentially give, extra permissions or donate the drug. So there are considerations with these drugs where we know what we need, but we don't always have access to what we need. That's the first thing. But on a theoretical level, we want antivirals, and we would like antivirals, first of all, with different mechanisms of action. There are antivirals that are better, that disrupt, for example, different parts of the viral life cycle. So if you could, for example, use, and this is the case with the treatment of other persistent infections, perhaps a combination of 2 antivirals, each with somewhat different mechanisms of action that target different parts of the viral life cycle, might make the most sense, and really getting at a persistent reservoir from 2 angles to make it not be able to continue to survive, if you will, right? So that's a consideration. With the antivirals also, another consideration is length of dosing, where in other chronic infections, and we mentioned several in the paper, for example, we talk about hepatitis C, which is a chronic viral infection. The standard treatment for hepatitis C is 12 weeks. And the early trials for hep C treatment were actually 24-week, often trials, or even longer extended courses of antivirals. And thus, the ability to dose the antiviral over time is important in the treatment of other chronic infections. Requiring that length of dosing does set some precedent that might be needed for the treatment of SARS-CoV-2 persistence as well. Part of the reason for that is that if the virus is, in a cell, so let's say, it's hanging out persistently, you often need that cell to die off, so that the virus can't get to the next cell, and the infected cell actually dies and the reservoir has more trouble moving to the next cell, right? That is more likely to happen in shorter lived cells. So for example, cells of the gut sometimes have shorter lifespans, and you get more cell turnover, which helps the antiviral kind of begin to clear cells that were infected and get in the way of the whole infection process. But in longer lived cells like neurons or even certain macrophage cells in tissue, those cells live for a really long time. So if they're harboring persistent RNA or protein, it's harder to get the virus out of those cells, which don't turn over as often, which is part of why that longer length of dosing can often be needed to account for persistence of infection in those longer lived cell types. So that's a major consideration with the use of antivirals. Another just example on that front would be there's a coronavirus infection known to impact cats in it's a persistent coronavirus infection, feline infectious peritonitis. And again, treatment of that persistent coronavirus in cats, again, it's 12 or more weeks of treatment of an antiviral. So you have to put that in perspective of the trials that have been run thus far in long COVID. So far, we've had antiviral trials of Paxlovid, which again, I think we should have run these. We need to start, we need to try, we need to test drugs and see what happens. But the trials of Paxlovid have been 10 days long or 15 days long. And I think one trial is 25 days long. That is short in terms of these other longer dosing periods that we know are needed for other persistent viruses. So that's a consideration. And then also, as David mentioned before, the tissue penetration of the drug. This really goes for all the drugs, not just antivirals. It's exactly what David was talking about before, is if the drug that you're giving someone can't get to the sites, that the person has a reservoir in. For example, if the drug doesn't cross the blood-brain barrier, it doesn't get into the central nervous system well, but someone could harbor persistent virus in the brain or central nervous system, that can be a problem as well, because the drug doesn't access the location where it needs to target the virus. So those are all considerations. David, I could let you talk about monoclonal antibodies, which are on the list as well. Are you sure? Okay, I'll just keep talking. All right. Okay, so actually,
you're on a roll. You're on a roll.
Yeah,
Keep going.
I can't talk about this for long periods of time. Many people who know me. So the next on the list, actually, we'll just go on the list direction, is that, and these are still don't really exist right now in a, okay, we can go get them off the shelf use, but antisense oligonucleotides or CRISPR-based approaches are being worked on in some labs for SARS-CoV-2 and certainly are being worked on, for example, to target HIV or other viruses. And what is cool about an antisense oligonucleotide is a little piece of genetic material that has basically the opposite sequence of the virus backbone that you want to target. So it comes in and it kind of like reverse binds to the viral RNA that's persistent. And by doing that, it neutralizes or inactivates it in a way that makes it not able to function anymore. And then hopefully the host cell, dies off and you neutralize the virus and you got rid of it. Same with the CRISPR-based approaches. And CRISPR is a tool that breaks up genetic material. So a CRISPR-based approach could target a therapy. And again, if the viral RNA is here, it could literally just break it in half, which would be great, right? Now, the main considerations there, though, are, again, these technologies are still more in development, and you have to think through how you use them because they have to be very well targeted to the site of the reservoir. So for example, if you're using an antisense oligonucleotide to try to bind and neutralize persistent RNA, you have to be able to direct that oligonucleotide as part of the drug to the site of that RNA. That's still a challenge in the field of people who are making oligonucleotides. So there are still considerations about getting these kinds of therapies to work really optimally. But it's the dream, in my opinion, and it couldn't, boy, should we fund this area of research, because being able to just get at the RNA itself or break up the RNA itself is the best way to just get rid of a persistent infection. That way, don't we have to worry about if we got it in a period of replication or a period of movement. We can just know that we can break it up. So that is part of the future where, we could put some really awesome funding into right now on the advancement of those types of therapeutic approaches. But then what actually, luckily, somewhat exists out there now are monoclonal antibodies. Although I think many patients in the long COVID community are like me and David, very frustrated that, their monoclonal antibodies are somewhat out there, but are to some degree sitting on company shelves and not being trialed in long COVID, or the FDA doesn't have programs by which people with long COVID could access them right now. And so, there are companies that want to get monoclonals involved in the space, but it's a thing, and it requires regulatory approval, and it requires actually running good clinical trials, again, to show efficacy, to show possibility, and all of that requires funding. So it's a process. But overall, a monoclonal antibody, and I think, some people might recognize them from just, acute COVID treatment. It's an antibody, literally. So when your body is fighting an infection, it creates antibodies. We all know this. And what this is really, it's just giving a person an antibody that, was created ahead of time, that's already targeted to go after the virus or its protein, right? And kind of infuses into the person the immunity that they might not otherwise have, and it helps them go after the virus. And some of the considerations for monoclonals are, though, that most monoclonals recognize viral proteins. So for example, the spike protein. So a reservoir that's, let's say, inside a host cell needs to have, the cell needs to technically kind of be expressing the protein, let's say spike on its surface, for a monoclonal to be able to recognize it and know to go after that cell. So a very hidden reservoir in which, protein is not being expressed on the surface of the cell might not be noticed by a monoclonal and missed. It's a consideration of monoclonal use. Also, the monoclonals, depending on which ones you use, are sometimes targeted to a specific variant of SARS-CoV-2. So some of the monoclonals were designed to go after the Delta variant of SARS-CoV-2, and some were designed to go after the Alpha variant, some Omicron, a couple. And that means if you're using that kind of monoclonal that's variant specific, you would need to know that the reservoir someone has is that variant. So in other words, a Delta monoclonal would likely only work against someone who has a Delta reservoir of SARS-CoV-2. So that's kind of a tricky process in trying to coordinate what reservoir someone has with the monoclonal, and that's a whole process. But what is encouraging is that more monoclonal antibody companies are creating broad spectrum monoclonal antibodies. And these are monoclonals that have been designed to actually have activity against most variants, right? In other words, they have broader spectrum activity that could neutralize, in theory, any type of variant of COVID. And those broader spectrum monoclonals, that's exciting. There, you wouldn't have to think so much about, hey, what reservoir is this? When did the person get infected? And maybe the broader spectrum monoclonal will have activity against any form of reservoir. So those are exciting drugs to keep in mind in this space. And many people, including myself, when we propose in this paper, do think that a great trial in long COVID that is long overdue, that we really are trying to make work happen, but we need our industry partners to really come in and help us with this. And there's some progress, is a trial of an antiviral and a monoclonal antibody. There you have two different mechanisms of action together. You're targeting the virus more directly with the antiviral. You're bringing the immune system in a bit, recognizing the spike protein in infected cells with a monoclonal. You have these two different drugs. This is a common combination in HIV, for example, treatment, monoclonals with antivirals, and in the treatment of other infections. So it would sure make sense to do that kind of combination therapy in the long COVID space and see what happens, right? So yeah, those are some of the options. And the last one, is still on the previous picture, but is that there's a couple at the bottom there that we can just summarize together, which are treatments that would just help support the immune system itself to go after the reservoir. So forget actually trying to get at the virus itself. What the other treatments do is say, oh, T cells could be exhausted in people, let's say, who haven't cleared the virus. What if we bring in a drug that makes the T cells more energetic? And then the T cells in and of themselves can come in, gain the energy to come in and clear the virus themselves. Same with potential, let's say, natural killer cell. You could bring in natural killer cells, up that function, right? So I love that approach. I think supporting the immune system is the future of treating chronic infection. In other words, bringing up a person's own system to fight the infection. And, I think that one of the only things that has to be considered there is that could potentially lead to a rise in symptoms in people who are getting that kind of treatment, because these kinds of therapies are done in cancer, where sometimes when someone has a tumor, we're able to engineer T cells that go after the tumor and attack it, which is what you want. But when the T cells come in and attack the tumor starts to get broken up and the immune system starts to fire, and there's inflammation, there's byproducts of the tumor. And that leads to what's sometimes called immunopathology, where the person can actually feel substantially worse, at least for a while, as that fight happens. So the same could be true here if you activate the immune system to go after a persistent infection like SARS-CoV-2. As it comes in, goes after the virus, tries to fight it, can get an increase in symptoms. So that's really important when considering the design of trials, because if we expect the therapy to make someone feel worse, we have to design the clinical trial to account for that, or everyone's just going to drop out, right? So that requires us drawing from the field of sometimes HIV or cancer to understand how teams are dealing with drugs that activate the immune system and lead to temporary increases in symptoms as part of what they do.
Thank you, Amy. I always love listening to you talk about this subject because you're just so great at explaining this. But one thing that I appreciated about the paper was just your analysis on the current completed and active clinical trials that are designed to prevent or target the SARS-CoV-2 reservoir. So there are several studies that you've outlined here, and then you go into the challenges of designing what the main considerations are rather for clinical trials that target the reservoir. And we've talked a little bit about these, particularly we've talked about eligibility determination earlier in the conversation. And you also just mentioned the combination trials, but there are other factors here. And I'd love to hear more about what you've talked about and what your thoughts are.
Yeah, I think, we have covered many of these, but on-site reinfections is definitely an issue. I'll be honest, there has never been, to my knowledge, a time in history where people are trying to run clinical trials to help people get rid of an infection that they keep getting so often, put it that way. Like, you could get other infections, but the rate at which COVID is circulating and infecting people, it's unprecedented. So obviously it can throw the data of a trial. So if we're, measuring people's symptoms, response to treatment, maybe even measures of persistence in someone in a trial, targeting the reservoir, and then they get infected with COVID mid-trial, and it ups their symptoms again in a different way, and they, for all we know, get another reservoir. I mean, this is obviously going to could skew the results of the trial. So it's just an ongoing challenge. No question that the staff should be masking with high quality N95s in these trials. The last thing we need is when people come in for the trials that they get COVID. There's some really straightforward things like that to prevent people getting infected during a trial. Many people in the long COVID community are very careful with COVID, understandably, since it's the infection that started their chronic disease. And we have to hope to some extent that at least some of those measures allow people to get reinfected, not as often in these trials. The one thing that, if you make the size of the trial big enough, and of course, a large number of people in a trial is always helpful, because that way you have, kind of a bigger group, so that if there are smaller events that happen, they're sort of somewhat diluted in the signal. You would actually hope that in a large group, I guess the same number of people, give or take, who are given the drug versus the placebo, the inert molecule in the trial, kind of get reinfected in the same rate on the two different arms. But we can't even totally account for that. So it's a challenge for the field, period. And then,
well, I think
yeah,
I think it's a challenge, but it's a challenge that we can overcome with good technology and good practice, right? To your point, we need to make sure that sites that are running these trials, at the very least, study staff are taking adequate protections. Environments are controlled with, things like far UVC lighting and HEPA filtration and clean air and a sort of a good rate of exchange of air in the spaces. I know, Amy, you and I have been talking about the idea of course, regularly testing our patients for evidence of infection, as well as setting up filters in the home that can actually detect presence of pathogens in the home of research participants. Because, again, to your point, these are such complex trials. It's a population that small deviations in homeostasis can cause large deviations in symptoms. And if we're not measuring all of that, we can get the wrong idea from our clinical trials.
Yeah, that's, I think definitely a consideration. And then I think, on the bottom right box, prevention of long COVID, one of the things that we think about, and this doesn't have to do with trials of long COVID as much as it does of just what we should be doing, is we really could use more clinical trials of more interventions during acute COVID to prevent people from getting SARS-CoV-2 to persist in their bodies at all in the first place. This really has not been done much. People are taking sometimes 5 days of Paxlovid if they get sick, but no one's tried longer coursing of antivirals in acute COVID to prevent progression to long COVID, or combinations of two drugs in acute COVID, again, to prevent possible seeding of a reservoir in the first place. So, because this is the maddening part of any condition, chronic condition initiated and exacerbated by a persistent infection, is that if you don't get the infection or you treat it early, you can potentially prevent the whole chronic problem. So we can't ever leave out that box in the corner, which is that we could do more trials to prevent people from potentially getting SARS-CoV-2 reservoirs ever seeded in them in the first place. So those are major considerations. And then I think the last, point of one of the bigger points of the paper that's really relevant to the field right now goes back to what David was talking about before, which is we talk about two main designs of long COVID clinical trials targeting a reservoir. The first design is what most of the groups thus far and most of the teams in those trials that you put up on that figure there are doing, which is understandable, which is recruiting in any patient with a long COVID diagnosis into the trial. And then hopefully, depending on the group, and we do this at Sinai in our trials. So for example, we're running a trial of Truvada, Maraviroc, in efforts to see if maybe there's an impact on persistent virus in some of those people. We're not sure, but we think it's worth a shot. We measure in participants at various trial time points. We look for SARS-CoV-2 proteins in their blood. We look for other metrics of persistence in participants, before they start the drug, in the middle of the trial, after the trial. And what that means is that even if the trial doesn't meet its primary endpoint, if the whole group doesn't basically get better on a group level, we can look at certain people who responded, like some of the people who got the treatment might have gotten a lot better. We can look at them and we can say, did that person have, spike in their blood? Did they have these other immune parameters? What were the biological characteristics of the responders? And that can help us understand that a certain subset of people in the trial were the ones that the drug was able to help. Then we can use that responder information to potentially design a larger trial, a second trial, where we only recruit in patients with those characteristics or markers into that trial, which is much more likely to meet its primary endpoint. The main thing to understand about those studies is if you're recruiting in everyone with a long COVID diagnosis, it's very unlikely that everyone has persistence, okay? Because there are, as we know, just other issues that can happen in people after COVID. So for example, the virus could knock the immune response down and Epstein-Barr virus could reactivate in someone. And now that might be that particular person's primary problem. The Epstein-Barr virus is driving their symptoms. If they're in this persistence trial, they might obviously not respond. And so these trials, if they don't meet their primary endpoints, we stress this in this paper, it doesn't mean a couple things. It doesn't mean that drug even failed. It doesn't mean that it can't work for some people, in other words. It doesn't mean that persistence isn't happening, which is the most annoying thing I'll hear people say. They're like, oh, this Paxlovid trial of 10 days didn't meet its endpoint, for everyone. Persistence doesn't exist. Well, no, I mean, there's all these variables that can be impacting what happened in that trial because you don't even know that everyone who was treated even had a reservoir, right? So these earlier trials are more signal finding trials than they are trials trying to make definitive statements in this field, right? If you want to move to a trial that's more likely to meet its endpoint, you have to recruit, and we discussed this is the second model of long COVID persistence trials. You have to recruit people on some measure of persistence. So for example, only people with spike protein in their blood would be included in the trial, or only people with T cells still responding to the virus would be included in the trial. That helps you narrow your group down to a group that is more likely to consistently have a reservoir that can be affected by a therapeutic. And that second design is the design that we'd like the field to now move forward with more. And that's why we're so interested in moving forward these different tests that can identify signs of persistence in accessible fluids so that we can better recruit and run that second type of trial design.
Thank you, Amy. And we are right on time for the 15 minutes of Q&A. So if there's, if there's anything else outstanding that you two would like to bring up before we dive into these questions that I've set aside, we can just get straight to that. Okay.
I was just going to say, I really just want to drive home a couple of the points that Amy brought up, which is that, when pathogens persist, they're different, and we don't need to, again, this should be a call, this live stream should be a call to action. We don't need any more evidence that pathogens persist. And historically, this has just been such a common tale of history repeating itself that I think that clinicians and scientists need to learn. One of my favorite sort of examples of persistent pathogens is something called a MAC infection, which I don't think a lot of people know about. But back in my days working in pulmonary critical care. This is back in Australia. This is something that we used to talk about a lot. MAC infection stands for Mycobacterium avium complex. And it's this, it's this mycobacterium that was kind of in the dirt and soil, and people would breathe it in when they were out farming or, in sort of rural areas. And it would sit in your lungs latent for 40 or 50 years until once you hit 60, once you hit 70, your immune system started to weaken just a little bit, and it would manifest as a persistent cough. So this pathogen, this bacteria that had been sitting there for 60 years, just quietly living and persisting, would suddenly become active and would make people symptomatic. But it was because it had been sitting there for so many years, it was also hard to detect. It had formed biofilm, which is, like these sort of salivary coatings that bacteria use to hide from, the immune system. So we weren't able to see it in our tests. So doctors would say to patients who had this terrible hacking cough that just wouldn't go away, but it was a dry cough, meaning it didn't produce phlegm, so they couldn't culture anything. And they would say, oh, this is a neurogenic cough. So it's like an anxious cough, or it's something related to, something neurological going on, but it's not a real cough. Or they would just say simply, you're getting old, of course you're coughing. Like that's what old people do, they cough, It in fact wasn't until the 80s and the early 90s when AIDS was so severe that MAC infection was one of the main opportunistic infections that was causing people with AIDS to pass away, that people started to realize, oh my God, this is a very common mycobacterium that's causing a problem. And then they started to see that even outside of the HIV community, lots of people had this mycobacterium. And to Amy's point earlier about, how hard you need to treat these pathogens, these days, now that we understand MAC infection, I say understand, people still get told that they're crazy because they're coughing. It's still not perfect in diagnosis. A lot of physicians don't know about it. But when you do get identified with MAC infection, on average, people will be taking antibiotics for 18 months to clear this persistent infection. So this is a bacteria that we know about. This is a bacteria that we have effective antibiotics to treat, but you still, once you have held on to this pathogen for 40, 50 years in your body, you still need 18 months of antibiotics to clear it. So I know that I took up a few minutes of question time, but I just wanted to share, I think it's a nice way of rounding out a really concrete example of how we have seen persistence occur previously, and how we've seen how much it takes to treat persistence once you allow a pathogen to stick around for years. And so, with that in mind, again, this is a call to action. The clock is ticking. We need to stop denying that this is happening, and we need to start coming up with all of the things we've been talking about in this live stream. We need government support to do it, because this is a huge issue that is affecting so many Americans, so many people around the world, and all of the effects that come along with that, the economic effects, the pain that it's causing families. We need these tools to be able to address persistence.
Thank you, David, for that very important historical context. It's very useful and important. Let's try to get into some of these questions. I've set aside a few of them on the side. And a lot of you had very long questions. So this is in two parts. WG asks, this is two parts. Could a hyperimmune response, very high antibodies to offending variant, be caused by continual antigenic stimulation due to viral persistence? Surely very high antibodies should have cleared the virus, question mark. Part two, or could there be pockets of persisting virus hidden from our circulating antibodies? Or could this paradox be explained by a personal mutation that our antibodies haven't adapted to?
If you have high antibodies still to SARS-CoV-2 protein, let's say to spike, there's a couple different explanations for that. It doesn't necessarily have to be a hyperimmune response, in that it could just mean that there are enough spike proteins still in you based on not clearing your reservoir, that the immune system keeps making antibodies and thus they're elevated. In fact, there are a couple long COVID teams that have found consistently elevated SARS-CoV-2 antibodies in people with long COVID, which suggests that could be happening. If they're super high, there might be some inflammatory response that's possible, but I'm not totally sure. I agree though, if you have high antibodies, you should clear the virus. It is possible then there could be some issue with the immune system. Either there could be an issue with the immune system's ability to correctly be clearing the virus with those antibodies, or the person could just be creating spikes so often that, the antibodies don't just remain elevated because that's what they should be doing, right? So there's just a couple different possibilities there for elevated antibodies. Yeah, I mean, exactly. There could be hidden pockets, if you want.
I think WG asked a bunch of really good questions and kind of answered them as well. The only thing I would add to it is exactly what Amy was getting at. Like a lot of the things that we've studied as a group, we've shown that folks with long COVID tend to have T-cell exhaustion and other forms of immune system exhaustion. And that doesn't just mean that the immune system is kind of tired of fighting. It also can mean that the immune system is highly active, but is no longer producing effective antibodies. So T-cell exhaustion can affect the ability to produce effective antibodies against, an offending antigen. So if you have T-cell exhaustion, but you still have these very high antibodies, it could be that, yes, your body is producing the antibodies, but they're just not able to be as effective as your body would want them to be.
Yeah.
Thank you. And let's answer this question. Does normal T-cell flow cytometry mean no reservoir? Or is there no conclusive test to prove viral persistence?
Well, I think if you mean like on a blood test, to the degree that we can test T-cell activity, which is actually pretty limited in clinical blood work, which is a huge frustration. A lot of what T-cells are doing are not tested well by labs like Quest or LabCorp. We need total innovation in that space. You wouldn't necessarily, first of all, even if you could understand certain parameters of T-cells in someone, sometimes using a method like flow cytometry, it's hard to know what it means with a reservoir, because sometimes the reservoir is tissue-based, and the T-cells that you grab out of blood aren't always in the same state as they would be if they were in certain parts of the tissue. So it's a consideration that T-cell activity in the blood doesn't necessarily always, and we're not sure, reflects sometimes T-cell activity in other body sites. To get around the sort of what T-cells are doing challenge in terms of persistence, there are some groups that have technologies, and I mentioned one before, that can actually identify if a T-cell is still responding to a viral antigen. So what they can do is basically just sequence the T-cell, like understand the activity of the T-cell to show that it's exactly still responding. It's called like a SARS-CoV-2 specific T-cell. That kind of test, which sadly is not available in the clinic right now, could tell you that the T-cell is still responding to the virus in a way that more conclusively suggests persistence. There's the same thing where people can use like other sort of T-cell receptor based tests, again, to show that the receptors of the T-cells are still responding to a viral product. So the biggest challenge there is that those tests exist in some research labs right now, but we can't order them. Doctors can't order them. So there isn't a test like that we can order on patients, or even, or putting them in clinical trials is very expensive. That's the other challenge, is we really need enough funding, because those tests are expensive to do right now in labs. So the trial would have to have a lot of support to be able to include that kind of test, which would be very valuable in it. And sometimes that's very hard to get the funds for.
Thank you, Amy. And we only have 3 minutes left to answer questions, but I've had an idea pop in my head that I'll just take the rest of your questions and get the answers to them, and you can follow us on socials to get our responses. But thank you for these questions. They're really great. Um, ah, to pick one to leave off with, um, William Gibb. For long COVID patients with very high antibodies to spike, what are your thoughts on treating with monoclonals? Could it be harmful to inject antibodies into someone with a very high titer of antibodies to spike, or could a monoclonal help by binding persistent spike, allowing the immune system to calm down? Alternatively, which other treatments could help in this scenario?
That's a really good question. And I'm not an expert in monoclonals. I think you literally just spelled out the two different possibilities, which is like, okay, you're increasing antibodies, but what if it could effectively neutralize the spike now? And then that allows the other antibodies to calm down, right? If you will. So I know that one thing that I've been told by someone who works at a monoclonal antibody company that I respect is that maybe the dosing period for the monoclonal in people who have higher antibodies could be lengthened out somewhat so that they're not receiving a super high dose in a short amount of time, which really could kind of exacerbate some of those issues. But they have a lower dose of monoclonal that's given over a longer period of time to sort of stretch out the ability of the immune system to kind of recalibrate those two sort of forces pulling the person in the two opposite directions, like the neutralization of spike, but the increase in the associated antibodies.
Do you think we have time for one more quick question? Okay, great. So let's do, Susan said, could you speculate about how trafficking of viral remnants could cause or explain PEM?
Yeah, there could be a number of reasons. I think kind of two main drivers of PEM that are under study are problems with the blood vessels. So if your blood vessels are inflamed or there's problems and you exert yourself, now the blood vessels can't recover, and that there's problems with perfusion and oxygenation that can make someone have trouble recovering from exercise. The spike protein, let's say, that could potentially, be sort of come off a reservoir, targets the blood vessels. I mean, SARS-CoV-2 has even been shown to directly infect the blood vessels. And in fact, we have two studies studying if the blood vessels are themselves sites of SARS-CoV-2 persistence. Or the foam cells of, well, it depends. That's in people with heart disease, but never mind. Even the blood vessels themselves, right? So you could have problems directly with the blood vessels that are part of a reservoir or associated with spike that could impact those sort of vasculature, blood vessel related post-exertional malaise type issues. Or it's important to understand that every virus must hijack the mitochondria of the cells that it infects in order to replicate. It just can't even gain any substrates to create a new version of itself without hijacking our own mitochondria to do that. So inevitably, a persistent infection will alter a person's mitochondrial metabolism to some degree. And so mitochondrial dysfunction, which could also be involved in post-exertional malaise, could be tied to a reservoir via some mechanism like that, although both those require further study.
Wow. And right on time, Amy. Thank you so much for all of your expertise. And David, of course, you as well with your clinical insights. I am going to wrap up this session for us. So
I didn't do anything this one. This is a very light lift for me. This is all Amy. Someone send her a bottle of wine.
Seriously. I mean, if I could give it to you through the screen, I would. And thank you everyone for your questions. Again, if I didn't get the chance to put your question on screen because of the time, I'm eager to get you those answers, and I will do so via our social media. So follow us at Core Sinai. And also, again, keep an eye out on our social and our website at coresinai.org for the launch of our infection-associated chronic illness resource suite launching this month. And this will include resources for patients, for providers, even for students and the general public. So we're excited to continue being a resource for you for all things IACI. And have a great day.
Thanks everyone.
Bye.
Cool. Bye.